This probably isn't going to be that smooth. It's our first time we've done it on such a scale, but I hope you can bear with it. Can everybody hear me all right?
Yes.
Yeah? Okay. Yeah. So we'll go... Okay. Right. Sorry about that. I had a big delay. So about half a minute later, I could hear what I just said. So I've taken my earplugs out now. Okay. So we'll dive straight in and go to the first session. So can everybody see the agenda on the screen? If you click on my screen, it should come up. It's also on the wiki. You should have been sent the link. Yeah. Okay. So briefly, an overview of EnteroBase. We have 4 public databases: E. coli, Shigella, Salmonella, Yersinia, and Moraxella. But we do have other private databases, which you'll see in a minute. So if you're interested in anything else, please let us know. With all the databases, we daily harvest all the short oil leads from NCBI and EBI and assemble them into genomes. And then these genomes are analyzed. So we assign 7-gene MLST, ribosomal MLST, and core genome and whole genome MLST. So ranging from assigning 7 genes to greater than 20,000 genes. We also annotate every genome using Proca, and depending on the database, we do other things. For instance, in Salmonella, we give it an in silico serovar prediction using a program called Sister. And in E. coli, we give it an in silico serotype, the OH group. And that was in-house developed by Zemin. So as well as all this data we get from the public archives, users can also upload their own genomes. And these can remain private for up to a year. Users can also search all the strains. They can download all the data and genomes, find the closest neighbors of their strain of interest, create and share workspaces, which are basically just groups of strains and analysis, draw trees to assess the population structure, and call SMPs. In the pipeline, we've got some more improvements. I've nearly finished custom views and columns, so you can add your own data and own fields. Also, people are interested if they've got a continue? Yes. Okay. Also, people are interested if they have a specific sequence. They want to know whether this sequence occurs in all our genomes and visualize it graphically. So I'm working on that as well. And we are constantly asked about antibiotic resistance prediction. So all these things are in the pipeline. Okay, so next we're going to move on to data privacy and usage conditions. So we actually have a page on the wiki for this. So if everybody wants to read through all this now, and then I'll test you at the end. So do you want to say anything about this, Nabil? Or
the main part is at the end, Appendix A. That gives you specific notes on the usage of things like the MLST schemes and so on.
Okay. So, right. So, and also briefly how to register and log in. I think most people will be familiar with this. You can always go to the wiki page if not, but just to say that you don't actually need to be logged in to just search and browse and download the data. However, you do need to be logged in to upload reads or create workspaces and analysis, and also obviously to edit metadata. This is mainly so we can keep, so if you create a tree, then we know who's created it, so you can come back and view it. We don't keep tabs on anybody. So we'll move on to the just general searching and browsing. So this is the front page, and you can see I'm logged in at the moment, so I have access to all our private databases. But we're going to go to Salmonella. Okay, and then to search strains. Okay, so this is just your basic search box. You can pick a field and an operator, you know, your basic query, and then a value. You can have multiple queries and combine with and or, and then you can group them. You can have and or. It's just the standard. So you can search on strain metadata or experimental data. So this is schemes or serotype prediction or even assembly stats. You can combine the search on the strain metadata and the experimental data just on a simple and or. The reason why you can't do complex joins is because the experimental data comes from disparate sources. So it's not a question of just joining a table in a database. Sometimes it comes from an API or somewhere else, so I can't do a simple join, but you can and or the strain metadata and the experimental data query. And that's what we're going to do now. So what we, under the serotype prediction and serobar, we're going to look for serobar called strain 4. Okay. And then under the strain metadata, we're going to check for anything that is not equal to St. Paul. There you go. So here we have in the meta. So this is the fields people have put in when they've uploaded the data. So we want anything that's not St. Paul, and we want to combine it that everything in silico prediction that did say St. Paul. Okay, and then just press submit. Okay, and you can see here at the top we have 89 rows. Okay, I'm just gonna okay, and obviously in the serotype prediction everything is St. Paul, but on the metadata that we've harvested everything is different. So you have to be aware of this, that the metadata is not always that accurate. We try to curate as much as we can, but obviously we can't do everything. So under the experimental data on the right, we can choose to look at different things. We could look at 7 gene. Okay, so for the smaller schemes, you actually get all the alleles. But in larger schemes, you only get the ST because it would be a bit crazy to have 3,000 columns. However, with the new custom views, you can then select which alleles you want to view. So you could create your own sub-scheme with your favorite 20 loci. That is coming very soon. So I just showed you a basic search on metadata and experimental data, and now I want to show you how you can view your closest neighbor of a strain. So there's many ways to do this, but perhaps the easiest is to get the scheme in the experimental data that you want. So in this case, it's the core genome. Then right-click on the strain that you want to find the closest neighbors to, and then go to find STs. Okay. So this happens to be ST37408, and we want the maximum number of mismatches. So we're going to allow 200 mismatches. So this is out of over 3,000 genes. We're going to pick all those that only have 200 or less mismatches with this strain of interest. Okay, so this will take a while. I think there's a timeout of about 60 seconds. So if it's a complicated search, you might get a warning saying that it didn't work, but you can try again. If it persists not to work, then please contact us. Okay. I promise you this worked last night.
So while we're waiting for that, if people are having any difficulties with latency and the speed is very slow, I'm seeing on the YouTube link it's much faster and much easier to watch. And there's a link to that in the chat box. Secondly, be there in the live chat for the YouTube video, and I can answer questions there. We have a question, Martin.
Yeah.
Is it possible to add columns to metadata, e.g. in my own workspace?
It will be in about a week.
In a week?
Yes.
In a week. Okay. Okay, continue, please. Sorry.
Okay. Okay, so now we have 674 strains that were within 200 allele differences of the one we picked. And you can see the number of differences in the left-hand column in the experimental data. Report on this. Okay, so obviously you've got one with no differences, which was the strain we started with, and then one with two differences, obviously quite similar. So you can sort by clicking on the column headers, but if you want to sort on more than one column, and you can sort between the experimental data and the metadata, you can click on the sort icon at the top of the screen, and you can sort on up to 5 different columns in order. Okay, so what I was going to do now was actually, seeing as we've got these strains, we're going to create a workspace. So we can just go to workspace and save as, and we can call it St. Paul test and save. Okay, so a hidden feature that not many people use is that you can actually change the settings. So if you don't like the colors, you can change them, and you can change the font. So lots of people like Comic Sans, so that looks much better, but it's up to your personal preference. And actually, when you actually save the workspace or resave it, it will memorize these settings as well. It will also memorize the column widths, and also if you show or hide any columns, this will all be memorized. So when you load the workspace, it'll be exactly as you left it. I'm not going to save this particular layout because people will get annoyed. So I think next on the agenda was buddies. So I've created this workspace now, and I want to share it with a friend. So if you go to tasks and my buddies, okay, you could start typing the name of the person you want. Yeah, that's my colleague Bill. I'm going to share this workspace with him. So I need to add him, and then on shared workspace, so I can click on this and share the one I've just created, St. Paul test. Okay, and then I can update it, and he'll be able to view that now. Okay, so what was next? So we've done general searching and creating a workspace and sharing it with a buddy. So next is uploading reads. Okay, so for this, I am going to go to our test database. So this is full of rubbish, and we can just put whatever we like in here. But as you can see, we have some eminent microbiologists using this at the moment, Cock and Pasta, and also some non-eminent ones as well. Anyway, let's move on. So if we go to tasks and upload reads, so if we just have one or two reads to upload, we can do it on the interface here. So if we go, first of all, we'll click on read files, and then we could choose our reads like this. Okay, and depending on the database, there's some requirements for metadata. In this particular case, all we need is the name of the strain and the lab contact. Other databases might differ. So now there's no red left, we can submit the data. Okay, so now we've submitted it, and we can see the reads are ready here to upload. So we can start to upload. So what I'm going to do, I'm going to start loading it and then stopping to simulate what would happen if something went wrong. Because what I see a lot of people doing is they'll upload reads and then the upload won't work, and then they'll go back and re-add the metadata and keep doing that, but you don't need to do that. So I'll just give an example now. So if I start the upload and then immediately navigate away from the page, even though it tells me I shouldn't. Okay, so I put the metadata in, but my reads have not uploaded yet. So what you do, you go back to the uploaded read page and to the uploaded reads, and you'll see here I've still got two awaiting upload because there was a problem and they didn't upload properly. So all I need to do now is find where they were on my local drive. So they were in test data, and you can just drag and drop the ones that didn't upload, put them there, and then you can do it again. So there's no need to go back and keep adding the metadata again. You only need to do it once. So now I'll let it go to completion. Oh, you see. Okay, you see this is failed upload, but I'm actually doing it on my development server, so there might be a problem there. But for this particular circumstance, it doesn't matter. Okay, so one thing I would say, if you have a lot of reads to upload, you can save the template to a local file, to call it upload test. Okay, so this is saved into whatever directory your downloads from your browser go to. It's saved as a tab delimited text file. I've got my Excel set up so it can open those directly by clicking. You might have to open it directly in Excel. So if I click on this, you can see Excel will open, and I have all the headers, so I can start filling this in. For instance, and then you put what you like in there, and then save it. Okay. Okay, then you can go and upload. So it was in. Yeah, so if we upload it now, yeah, so we can see it's uploaded. So I think people use a script or something so they can associate all their FASTQ files with the appropriate metadata, and then they can upload it. Okay, so that was uploading reads. Editing metadata. So if we go back to our workspace that we created earlier, so you could load the workspace from the species homepage, and you can see here, this is the one. So we will load this. Okay, so this dialog here is just if you want to add a description or links, and it also shows the trees and projects associated, SMP projects associated with the workspace. So if you own the workspace or have permission, you can actually type in description or what you want. Again, we're going to sort. So you can see here we've got some mistakes. These should be some poor, and we want to change them. So we have to go into edit mode. Okay. So because I'm administrator, I've got a permission to edit all records. Normally you would only be able to edit records that you own, i.e. you uploaded or that somebody shared with you. If you want to edit other records, please let us know and we can give you permission. So you can either type directly into the box. Yeah. Okay. It turns yellow when it's changed. Or obviously this hasn't got not as many features as Excel. So what you could do, you could save to local file as before. Oops. So again, you'll go into the downloads folder of your that your browser uses, and you can open it. So it's just again, it's tab delimited text. So yeah, so if we go to ones we want to change, and we can change them all to St. Paul, and now we save that. Okay. Right, so when you're in edit mode, you get these extra icons here at the top of the screen. So if we click on the upload file one, we'll be just have to upload the file that we just changed. Okay, and ask us whether we want the barcode or accession as our key identifier. Just put barcode unless you've only got the accession numbers there. So, okay. So this will take a few seconds because it's querying the database, and then it should come back with any changes. Again, it worked last night. Okay, try it again. Oops. Oh, there you go. I think the network might have been slow. I do need to put a dialog box in saying that it's pulling the data, but as you can see, it has synced it with the Excel sheet or text tab delimited text file that we just uploaded now. So all the St. Paul's have been changed. When there's any yellow changes like this, they're not actually committed to the database yet. You have to press this icon at the top here to upload the changes, and then the database will be changed. I'm not going to do that at the moment. So basically, if you have a lot of changes to make, easier to make them in Excel and then upload them. Okay. So, do you want to carry on, Nabil? Do you want to say anything? Because I'm already in session 2. Shall I just carry straight on? Any questions?
Yeah, we'll take some questions at this point. If anyone wants to just pop it in the chat or something. No?
Okay. So, next on the agenda was monitoring job progression, but I'm actually going to do that later on when I start creating trees, because then we'll have actually something to look at and see their progression. So instead, I'm going to talk about what are Uber strains, and probably the best way to demonstrate this is to have a look at a search. Yeah, I think the network's a bit slow, isn't it? Try. Yeah.
Jump to the main side.
Yeah, I am. I'm going to the main side now. Okay, that's it. Yeah. Okay, so we will. Yeah. So we'll do a search, and we're going to search for the LT2, which I believe is the K12 of Salmonella. So. Okay, so there we go. We have one strain. And the Uber strain column is here. And okay, and as you can see, the Uber strain is exactly the same as the barcode. Okay, so this is the super strain. To explain this further, if we go back to the search, and now we're going to click on show substrains, and we'll do the search again. So now we have a total of 30 rows, but only one is showing. And the reason for this is all the rest are substrains of LT2, i.e. that other reads or things that people have uploaded, but they're all from essentially the same strain. So on our Uber strain, if we click on the triangle, we can expand this, and we can see all the other strains that are, in fact, LT2. So normally these would be hidden. So you can see all the substrains, and if in edit mode you can actually create sub and Uber strains. Again, you have to own the strains, but this is a way of sort of sorting the data so that when you draw trees, you don't have 1,000 of the same thing because they're all K12 or LT2. And normally you don't have to worry about this because on normal searches, you'll only get the master strain. But just to be aware, if you ever wondered what this Uber strain column was, now you know. Okay. So downloading strain metadata, experimental data and genomes, this is fairly straightforward. So if we just look at the latest 200 to be uploaded. Yeah. So yeah, we can see some were uploaded last night in the short read archive. So you could save anything that's in the table to a local file by just saying save to local file. Okay, and it will save it as is, so you'll get the experimental data, in this case the assembly statistics. That's all there is to that, really. So you could download assemblies individually, or you can select a few and then download them. This icon here. Okay. Other files you can download for annotation. So these, because these are brand new, they haven't been yet sent to the annotation pipeline. Yeah, but the so these have been annotated, so you can download the annotation files. And again, if you want to download loads of them, you can right-click and then download all the selected ones. Okay. If you want a lot of assemblies, or if you want a lot of metadata, then obviously the best way is to go through the API, which is what Nabil is going to be talking about this afternoon.
Martin, I have a question.
Yeah.
What kind of annotation do I get?
Okay, you get either GenBank or GFF format.
So how are they generated? What are they?
Okay, so this is just generated by Procker. Okay. So you can load them into Artemis or another suitable program. I think Artemis takes both GBK and GFF format. Okay. Right. So now
we have a question.
Yeah.
I previously had a problem with Procker GFF files. They were not full, did not contain sequences. Is it fixed now?
Okay. I don't think they do contain the sequence, just the annotations. So,
so the way I would interact with that is if you go back to EnteroBase, Martin.
yeah, okay.
I would download the contigs, which is under experimental data, assembly stats. Download the assembly, so that's all of the FASTA files. And I would load that into Artemis and then overlay the GFF on top. The advantage of this is that Artemis will correctly handle the multi-FASTA. Otherwise, with the GBK, I think it has some problems sometimes. It doesn't automatically concatenate all the sequences into a single sequence.
Okay. So,
we have another question. What kind of de novo assembly is performed?
okay.
Yeah. So the assemblies that we're talking about that are used to drive all of the subsequent analysis, we use spades. We have our own quality control and pre and post assembly that we use to make sure there aren't any scaffolding errors or anything like that. But yeah, essentially it's spades assembly. Another question, is it possible to delete your own data after it's uploaded?
No, not at the moment. You'd have to email us.
Yeah.
We have done that for people in the past who uploaded stuff they shouldn't have, but we can delete it straight away.
Okay. I think that's all the questions. Chime in with questions as we go. We're running a little ahead of time anyway.
Yes. Okay. So I think next on the agenda was minimal spanning trees. Do you want to say anything about them, the bill, about the base, how they're made, or I think everybody knows?
Probably not, but
Okay.
let's show one.
Okay.
And then
Well,
we can explain it.
Okay. So we're going to go back to the workspace that we created. So that's loaded. So when you create a tree, you have to make sure that the scheme that you want in the tree is displayed in the right hand side. So for example, if I had assembly stats and I tried to create a tree, it would tell me that it can't. On the other hand, if I went to the core genome MNST and now try to create one, that's fine. One thing I've noticed as well is that people will try and create a 7-gene MLST tree that have all exactly the same ST. So they'll just end up with one node, and I think Zemin's program crashes and gets very cross and doesn't work. So when you start creating a tree, you have this dialog here, and it'll actually tell you the number of nodes that are going to be present in your tree. And if it's less than 5, it gives you a warning and says probably not going to be very good. So you can give your tree a fancy name or just rely on the default one given, and then just press submit. Okay, so you get this pop-up window. If your browser doesn't allow pop-ups, you'll get a little warning, and it's best to allow pop-ups from the EnteroBase site for this to occur. So the tree creation goes on the server, and then this page will actually poll the server every 20 seconds or whatever to see whether it's done. You don't need to keep this page open. You can come back later, which is what I'm going to do. So what I'm going to do now is check on how this job is doing. So I'll close the window, and I will go in tasks. I will go to show my jobs. Okay. Okay, and we can see that at the top here, this job is running. So if you click on the I here, you get information about that job. I haven't formatted it properly. This is what I get back from our colleague Zemin in his job server. So if we have a look, so if we look here at the bottom, we can see it is complete now. Yeah. Okay. So if we were to refresh this page, it's yeah. Okay.
Oh, we have a query, Mark.
Yeah. Yeah. Okay.
If you make a tree
Yeah.
from public data,
Yeah.
can you then show it at a conference without the permission from the owners?
Well, we have this discussion all the time. To us, whatever's in the short read archive is public because it's a public archive, but people from the Sangha get a bit cross because they say you can't use it until they publish on it. So I would make sure that you contact the people who put the data up there in the lab contact if you're intending to do that. Would you agree, Nabil?
I would agree with that. Full description of what we recommend in the terms and conditions of EnteroBase. The short end of it is we don't check if all the data is what people are happy with. Some institutions don't mind, some institutions do. That's why we provide the lab contact, and if you want to present it, you should probably just double check with them. If it is in a paper, then And if you find the paper, then there's no problem, obviously.
Yeah. Okay.
I think most people don't mind for trees after our interaction with a lot of data uploaders, because it doesn't say anything specific about your data. But I would always check with them.
Okay, so we have a problem here, but hopefully it's not unresolvable. So I sent the tree off. So when we query the server, we can see that it is complete, but the EnteroBase is telling us that it's still queued. Now this can occur occasionally so that the callbacks get missed or whatever, but every hour any stale callbacks will get picked up. But at the moment we can't really wait an hour. So what you can do is, if you right click on your jobs, you can force reload. Okay, so now that's forced to reload. So it's got all the data from the server and updated EnteroBase with it now. As I say, you shouldn't really have to do this if you're not desperate, because there's a script that runs every hour that tidies all this stuff up. But if in an emergency, you can do this. Okay, so now if we go back to and load the tree. Okay. It hasn't come up there yet, unfortunately. So let's just. Okay, so for some reason the tree hasn't appeared. I'll have to look into that. However, I had last night I did create one, as you can see. So we will load that one. Okay. Okay, so this is one I created last night. So you can see it's formed a few clusters. You can color it by any of the metadata. So the algorithm that tries to draw the tree tries to maintain the correct link lengths between them. A lot of algorithms, they tend to space the tree out. So the link, although it looks better, the link lengths aren't accurate. So you can see clusters that aren't really there. To demonstrate this, if you go to the layout, you can actually unfix everything. So this then allows the algorithm to take over. And so you can see now it's spreading out. So it may look a bit better, but if we look into it more deeply, okay, so we'll fix them all now. So you can see that looks a lot better. But if we examine the link length, so the lengths between nodes are the number of allele differences between those two groups. So if we zoom in, we can see here there's a difference of 1, and here there's a difference of 4, but they're about the same length. And again, 5 here is the same as 1. So if you do this, you'll get better. It'll look better, but you have to be aware that the link lengths might not be that accurate. You can correct them again. You see, jumped in. So as I said, you can color it on all the metadata, but you can also color it by any of the experimental data. So for instance, in this case, we're going to color it by the Atman 7 gene data. So you can choose anything down here. So we'll click on plus. Okay. So now this has been added. So we can go up here and we can color it by ST. So you can see the majority is ST27 with a few exceptions.
So I have a question on that.
Yeah.
Okay, there's a couple. Firstly, I think the question is,
Hmm.
I think I read you may be adding AMR.
Mm.
Is this coming soon in the pipeline?
Not that soon. We have discussed this, but we haven't actually done any coding for it yet.
Will you show the custom coloration?
Yes.
Okay,
Yeah, I'm on the trees, yeah.
on the tree.
Okay, yeah. So, talking of antibiotic resistance, what you can do is you can colour it by allele data. And I remember reading somewhere that Jaya A is confers some mutations in GRI confer nalidixic acid resistance. So we can colour this tree now by the Jaya A allele. Okay, so it's just loading the data in. Okay, so you can choose any gene from the whole genome scheme, which, yeah, there it goes, which is about 20,000 genes. So here you can see there's a group here that have a different JARA. So you can colour it by allele at the moment, but we haven't got specific antibiotic resistance. So any changes you make in the tree, whether it's moving the branches round or adding new data, you can save. So you just go to save layout, okay, that's been saved. And then you can reload the tree. And it'll come back exactly as it was before. Okay. You can also add your own fields. So we could add, say, my clusters. Okay. And then you can select some and add a cluster A. Okay. So, but you could also choose the color. Okay, yeah. So there's a bug in this. The first time you do it, it won't work, but I have corrected that bug on my development server. So you can give it a different color. So yeah, that's it. And again, if you save the layout, okay, and then reload it, okay, it should, you can go back to my clusters, okay?
Okay,
Right.
so is that the end of that bit?
No, I've still got a bit more. So another thing you can do is, if you're interested in some of the strains, you can select them and you can create a link with the main table so we can load selected. Okay, so if I go back to the main table here, it will be load those strains in, yeah? Okay. So those 54 strains that I selected in my tree are now uploaded into the table. You can also then select some here, okay, and then go back to your tree. So if you clear the selection and then we highlight selected, you can see these are the 4 that I have just selected in the table. Okay. So that's, again, it should be in the wiki. There's lots of different things you can do to adjust the layout, etc. But you can play about with that in your own time. And of course, you can download the SVG as a diagram. Okay, that's it, yeah. Nabil,
Okay, so we have some questions that have been coming in.
Okay.
The first one is, I'll just read it out because people in the stream people on YouTube can't see the questions in the stream, so I'll just read out the correspondence. The question was, are recombinant regions in the case of CGMLST removed? Yes. The combination was, we tried to pick loci for the CGMLST scheme that are not recombining. But as you continue to add more genomes, those new genomes may have regions of recombination, which we do not clean up. So you may have some of that recombinant signal in your CGMLST tree. However, in the comparisons we've done of SNPs that are cleaned up for recombination, for data sets that we are fairly certain about the genealogy, and we visited with CGMLST, we find that CGMLST is quite robust to recombination. Secondly, the question was, is it the same for mapping-based SNPs? So the SNP trees are not filtered for recombination. That is the core genome SNPs according to the reference you define. We don't filter for recombination yet. I'll also clarify the sort of the fair usage about using other people's data on EnteroBase. So this is again covered in the terms and conditions of EnteroBase. We can't police everyone's usage of other people's data. Not even any CBI or EBI tries to enforce it. If you read their data usage, they simply say, which we also agree with, we are a conduit for sharing information. We provide it in good faith. We hope that if you see data that is interesting, you would contact the submitter and create a meaningful collaboration with them. And so we had some ones on the main chat, on the YouTube chat. Is there a cutoff for FASTQ file? So using data that's put into EnteroBase. Yes, reads will be trimmed based on their qualities using sickle, and only a maximum of 1 gig of base pairs of bases will be used from the spade assembly. And in regards to the question about gene typing, so what Martin has just showed you is if you can find a gene that's associated with AMR, you can create a custom scheme of your own using those loci within the framework of the whole genome MLST. And you can color code that onto your various trees as well. But we do not have an explicit scheme defined as yet. If you're interested in helping us develop such a scheme, we'd be very happy to hear from you. That's right, you can continue, Martin. Yeah.
Okay, we're going along quite quickly, so there'll be plenty of time for questions. So the last thing on the agenda was the SNP trees. So what I'm going to do is select these ones here, okay, and then load them into the main table, okay. And now I'm going to create a SNP tree from these. There was a good argument the other day between minimal spanning trees and calling SNPs. So there was a very good fight. But I think that SNPs obviously better resolution at the very minute level. But when you're looking for a broader range, then MLST is much quicker and better. But anyway, so we're going to now, with all the data in here, so there's 62 strains, we're going to create a tree, a name, okay? And then the only thing we have to do is choose a reference. So best to give it a complete genome, but in this case we haven't got one, so I'm just going to give it the first one here, and then we're going to press submit. Okay, so it's been successfully submitted. Okay, so these jobs then run in the background on the server. Again, we can go to the jobs and see what's going on. Okay, so you can see. So last night I created the same tree, and last night the first job that was sent off was this ref masker job here. So what this does is look at the reference for any repeat regions or difficult regions that are really difficult to call SMPs, and these are then will be masked out. Next, it calls a ref mapper job, and this is actually so the SMPs are called. Yeah. It's okay, so we've just got a question about strain names for text. Yeah, that is. So there was this problem before, and I think on the latest merge with our source code, it got expunged again. So I need to add that back in. But I will update the changes to the production server today. Yeah. Okay. Right.
But I'll also clarify one thing Gemma reminded me. When you're generating snip trees in EnteroBase, while we don't handle recombination, we do a light filtering of repetitive regions.
Yeah.
Adding that.
Okay. Right. So yeah. So when I created this tree last night, the ref masker job was sent out, and also the ref mapper. So we call SMPs based on the actual assembled contigs, not on the short reads themselves. It's not a mapping approach. So the contigs are mapped against the reference, and then any differences, small indels or SMPs, are picked up. So this is what happens in the ref mapper. Then all those positions are fed into another program, which will work out. Work out the consensus. For instance, if at a particular position that area is missing in greater than 10% of the genomes, it won't be called. And also any mass regions will be excluded in the ref mapper matrix job. And then finally, when we have a matrix of SMPs and their positions, this is sent to a matrix phylogeny here. And this job just creates basically a RAXML tree for you. So today, when I called it, it didn't send off a ref mapper or ref mapper matrix job because these are all cached. So all it needed to do was to do the mapper matrix and the phylogeny.
The upshot of this is if you're generating the same tree over and over again, just adding small increments of data, small increments of taxa, that will run. The first one will take a while, but the subsequent ones will be faster.
Okay, so we shall load the tree that I created last night. Oh, hang on. Right, yeah. So this is actually the tree that I've created at the moment. So no wonder it's not ready. Sorry. So the one I created last night, I should name them better, shouldn't I? Is actually in training and support test. That one. Okay, so.
Before I forget,
Yeah.
someone asked, can a user create a user's in an Uber strain? And if so, how? You didn't demonstrate how to create your own Uber strain. So we'll do that after.
Yeah, okay. I'm having problems loading this one.
You have to change your desktop button.
Yeah, I will. What's that one not loading? Oh well. Pardon?
Perhaps it's finished.
No, I think there's a.
No, my network is quite slow.
Yeah, no, I think there's a definite problem somewhere. Yeah, sorry about that. I can't seem to reload the tree at the moment. Let's see whether no, that's very strange.
Just loaded in your screen.
None of them are currently loading at the moment. Yeah.
You have screenshots in the wiki too.
Not as such. So, do you want to?
Explain of the overstem. The meanwhile, how to create an overstem.
Okay. Do you want to do that, Nabil, while I try and sort this out?
Okay.
So I'll stop sharing my screen. Yeah. Oh, yeah. Have I stopped sharing it now? Yeah.
Yeah.
Yeah. Okay.
Okay, I'll just switch over to mine. Okay, so I think everyone should see my screen. Yeah, you can see my screen?
Yeah.
Okay. So how to create my own Uber strain. Okay. So I'm just going to demonstrate it using some strains that don't make sense, just whatever's in front of me. So the first thing you need to do is get into edit mode. Yep. I will, so it's under here under tools, which is add to Uber strain. So I click that. It'll say please select the master strain. So that's the top level strain. So it would be the best LT2 genome in the universe, or the one you trust the most, or the one with the best metadata, and you select that one. You press OK. Then it'll ask you, please select the substrains to add to that. So then you select those, you press OK. It'll confirm with you, and then you press, it doesn't update it immediately, doesn't it?
No, you have to update it. Yeah.
So it's now done it on my screen, and you can see that I've paired these three strains with this one at the top. This has not been updated in the database yet. It's supposed to be a point where you can check what you've done. And to confirm it finally, there's a button up here, upload changes. Now I don't want to upload these changes because they don't mean anything, but if you press that, it'll send them off and it'll give you a notification that it's fine. Now, users, you as a user can use it and do it on your own data that you've uploaded or that your buddies have uploaded. You can't do it for random public data like these records on this page, unless you are a curator of the database. Okay.
Okay, I've sorted it now.
So okay. So you share your screen.
Yeah, okay.
And I'll switch back to you. Let's see if we have any questions. Okay, so there's some bioinformatic questions. What pipelines and/or tools do you use for SNP calling? So Jedman has answered this. The SNPs were called against a single reference, which is one you as the user specify. All other genomes, so these are the contigs, will be aligned using last, LAST. All SNPs in repetitive regions will be removed, and then the remaining SNPs are used to construct the tree in XML. Okay. It's all assembly based. We don't use BWA or mapping. We do check the base quality of the assembly by mapping the reads back onto the assembly after it's done for each genome that is processed by EnteroBase. And you can download a FASTQ file that shows you the quality scores for each base.
I'm showing that in a minute.
Hmm?
I'm showing that.
Oh, you're showing that in a minute.
Okay.
And yeah, that's it for that. So I'll jump back to you.
Okay. Can everybody hear me now? And so I don't know what was going on there, but I've just loaded the tree that I created last night, and you will get two windows. One of them is a JBrowse instance. So this is a lightweight viewer, a bit like Artemis. And so this will have the reference strain and any annotations that the reference strain has. So this will be the Proctor annotations. If it's our complete genome, it'll have the GenBank annotation. It'll also have any scheme that was generated. But what Nabil was talking about was, well, I've got here, it's actually assembly errors. But so this is when the genomes are assembled, by mapping the reads back, we work out the quality at each position, and you get like a FASTQ file. So these are just the FASTQ values. You can see in some regions, few bases are have got very poor Q30 values. So if you have SMPs in these regions, I think they should be filtered out. So the annotations that with this one are, so the Proctor annotation, okay. So you can zoom in and out. Yeah, and also the whole genome. So they will be similar, but not identical. So the whole genome scheme, not every gene is called. If there's multiple copies or it's repetitive, then it won't be called in the whole genome region, but obviously Proctor will call it. So improvements I've got to make to this are, you can view the SNPs, but only from one contig at a time. So I need to stitch all the contigs together. So for instance, you can, at the bottom track, you can see there's two SNPs here. But what would be more helpful also is that you, for each strain, you'd also be able to view where the contigs map to the reference. So in the SNP tracks, I want to overlay where the contigs map as well, because sometimes you get to regions where there's no SNPs and people don't understand why. But it might be because there was no contig mapping there, or it might have been they were masked out in the because they're repetitive regions. So I also need a track which shows the masked regions as well. So this is still in development. So you also get a tree. Again, you can alter the scale and zoom in and out or whatever. So the way you add data to the tree is exactly the same as you would with an MS tree. So if we go back to our favorite gene, then you can just add it in exactly the same way. And you can see here that probably because it's due to antibody resistance, its homoplasia occurs in different parts of the tree here and here. Okay. Also, I'm not sure whether this is uploaded. If not, I will upload it tonight. You can also, there's a link to David Anderson's MicroReact. So you can click on this icon here and it will create a MicroReact project for you. If you've already created one, then it will just go to viewing it. So again, this is one last night. Okay. So yeah, you can see that these are all some poor strains from across America, and you have a nice map and stuff like that. If you used MicroReact before, you will, it's quite easy to use. Okay, so again, exactly the same as the minimal spanning tree. You can select nodes and then you can load them into the main table. It's a bit slow, but they should load in there. So I think that's about it at the moment. Lots of times for questions.
I'm sure everyone will appreciate early knowledge.
Yeah.
Basically, question. How to see?
The question is how to see number of SNPs between the strains and bootstrap values. I haven't shown how to download the data, like the NUIC file.
Okay, yeah. So you can download the NUIC file or the actual matrix of the links at the bottom of the screen here. So so you on the branch labels, I don't know whether Zemin can answer this. You do get figures, but I'm not sure what they correlate to. If you go back to the JBrowse, you can see the number of SNPs compared to the reference for each strain. In the tracks, it's the number in brackets. Yeah, agreed. It would be a good idea if it makes sense to overlay this onto the tree and have that in the branch lengths instead. As far as bootstrap values go, Zemin said they don't make a lot of sense for this kind of tree, but I'm not an expert in these matters. So I think Zemin can answer that question. So, so again, the data's loaded now that I selected before, and it's a two-way interaction. So you can select things in the tree and then highlight them in this tree here. So it goes back. So you can see they become highlighted now, the ones I selected in the main table. So as Nabil said, you can download the NUIC file and put it in another tree program if you want. Or if you're a bioinformatician, you can download the matrix and do some analysis on it.
So the matrix is the SNPs?
Yeah, the SNPs. So it just has the strains and then the SNPs at each position. Yeah?
So x-axis, the rows are the strains, the columns are the SNPs at certain positions.
Yeah.
So from that you can regenerate the tree anywhere you like.
Okay. Yeah. Okay.
Sounds great.
Pardon?
Gemini sounds really
Okay.
The other way, the other screen.
Okay, I'll leave that out. So in response to branch lengths and bootstraps, the branch lengths were the substitution rates calculated in maximum likelihood. So that's what Martin was showing on the tree. We are currently not running bootstrapping. Bootstrapping only works for uncertainties due to lack of data. So, so because in a tree like this you have several thousand SNPs, you don't have uncertainties due to lack of data. Your uncertainty is due to recombination or non-specific matches which will not actually be tested by bootstrappings. And so having a bootstrap tree, thinking it's confident, is kind of meaningless. Question is, can CGMLST allele sequences be downloaded via the GUI? Oh, you haven't shown how to download schemes in the browser.
Okay.
If you want to show that.
Do you want to show that?
No, you've got it. It's just there.
Okay. Well, you better. So this one here, yeah, download schemes, yeah.
Download schemes. So we make all of the every day we create a downloadable compressed file of all the allele sequences for each locus and the ST profile for each scheme, and that's available on the tasks on the download schemes. Okay, and that includes 7-gene MLST, core genome MLST, and whole genome MLST. We do not provide our MLST to the service. That you have to get from University of Oxford. So to download the scheme, you simply select from the drop down at the top there, so which says WGMLST at the moment. Can you change that to 7-gene?
This one.
Yeah, that one. Make that 7-gene.
Yeah.
And just do download ST profile.
Yeah.
And then that'll have all the allele numbers and the ST. And if you go to filter or clear filters, and you change the scheme name and make it, contain, no, make it 7-gene again.
Oh, 7-gene.
Yeah, 7-gene down the bottom. Yeah, filter. So those are the familiar 7 loci, ROC, DNA, and so on. If you select, click one, or you can click all of them, and then do download alleles for selected loci. There. You'll get 7 files, 1 for each locus, and you can see them loading down the bottom. Sake, histi, Ture, and so on.
Yeah. Okay. But I agree it would be good on some of the trees where you could just click on and it will download the allele sequence. So that's something I can think about adding rather than having to download everything if you're just interested in 1 or 2 allele sequences.
Concatenate.
Yes. Yeah, who's asked that?
That's good.
Yeah. Okay.
So what's the question?
Some people wanted concatenated genes. So
For the 7-gene scheme.
Oh, I see.
So that's another possibility. But I
At
think
least from the
yeah.
appropriate.
But I think that would be a bit tedious if you tried to concatenate the 3,000 genes from the core genome.
The 7-gene.
Okay. Okay. So yeah, please ask lots of questions in your own time, and we'll try and answer them. And I'll try and out any problems that popped up today as well. Okay. So is that it?
I'll check the various feeds and see if anyone's responding and any questions. Cool. I think that's it. No one's feeling any questions. Oh, that's nice.
Okay. Finished?
Yeah.
I think that's all the content we wanted to cover. That'll give you a basic understanding of how to do things on EnteroBase. Yeah, so the YouTube channel doesn't have any questions either. So I guess we will adjourn this session for a break. After the break, I will be showing how to interact with EnteroBase programmatically using scripts, using the API. This sort of requires, if you're not interested in programming or working, or you don't like computers very much, this session probably isn't of interest to you. But if you're interested in that, please join us back at, what did he say, 1:30 on the agenda?
The agenda.
Yeah, that's it.
1:30.
Yeah.
So in 1 hour. So we've got a 1 hour break now, basically. And at 1:30 we'll resume again, and I'll be talking about the API and programming with EnteroBase. Feel free to email me any questions. I'll send a response, and I'll also append it to the next. If you're in the next session, I will send you a separate link for that session, because we're sort of making these separate blocks. All right, thank you very much for showing up. Have fun using EnteroBase.